Thank you for joining us for today's webinar, How Multiplexed Microarray Technology is Revolutionizing Microbial Testing in Cannabis. It is presented by Milan Patel, co-founder and CEO of Pathogen DX. My name is Judy O'Rourke, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone this event is interactive, and we encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Milan Patel. I will now turn the presentation over to him.
Thank you so much this morning. I'm pleased to be presenting how multiplexed microarray technology is revolutionizing microbial testing, as part of the LabRoots virtual webinar. I'm Milan Patel, co-founder and CEO of PathogenDx. For the better part of 140 years, the world's been basically focused on microbial testing, and what I'm about to present is really the future ahead, and it's now. And things are simply about to change in the world of testing. When you look at the cannabis testing industry, you will notice that it's ripe of contamination. It's plagued with a lot of contamination issues across the entire supply chain, and up to 30% of the industry has purity and contamination issues starting from the growers, where labs have detected contamination of yeast and mold, bacterial and fungal, that is impacting the health and safety of the public as well as the consumers in that market. And as we all know, there are over 30 or so states that have legalized medical marijuana, and 10 or more that have already have adult use, and several more that are currently on ballot. What these states have done is basically put in a regulatory system that ensures that there is mandatory compliant testing of cannabis that ultimately gets to a patient or a consumer. A microbial test is one of a handful of tests that a certified independent test lab will be required to do in a legalized state, and it's the one that is the longest out of all of the tests that are conducted in a cannabis testing lab. It normally takes 2 to 6 days for results to be delivered to a cultivator or a grower. All other cannabis tests take less than 2 hours in terms of results. So growers are waiting for results from the microbial test, trying to get their product to dispensaries so that consumers and patients can have accessibility to the product in the marketplace. Just to take a few steps back, we all know that microbial testing effectively is the testing of bacterial or fungal contamination. And for the better part of 140 years, the world has pretty much used the classical Petri dish approach in identifying if a product has been contaminated with E. coli or Salmonella. The classic examples that we are all familiar with is last year, the romaine lettuce outbreak, where E. coli effectively infected the product, and a lot of product had to be recalled or scrapped off the shelf. And previously, we also had examples of Chipotle. The issue with the Petri dish technology, although it's being used for over a century is that it's simply too slow. As I mentioned earlier, it takes anywhere from 2 to 6 days. It's significantly variable, where the result can vary up to plus or minus 30% on either side of a fail pass threshold. And lastly, it's very costly. It requires a lot of waiting time and labor time, which is still costly in terms of getting a result to the growers. What PathogenDx has created is a game-changing, disruptive technology that allows to test for multiple pathogens all at the same time in a single reaction. You can see a slide on this, a glass slide on this presentation slide with little circles on them. These particular circles have the capability to test for at least 4 to 5 dozen pathogens per sample, delivering significantly faster turnaround time of results, down to 6 hours from 2 to 6 days. It gives labs a capability to process more samples per day and not have a bottleneck in getting bogged down with a lot of petri dishes. The platform allows for what is called content scalability. And what that means is if there is a strain of pathogen that needs to be added, the technology can quickly add that within 60 to 90 days to be able to test for the latest bacteria or fungal organisms that are pervasively infecting our food or water or supply systems. And in addition to that, it allows for a very simple and easy testing process for the labs. There's automated controls and built-in capability to make it user foolproof. And so it allows for less human intervention in determining whether you have what the result is. And lastly, it basically effectively can result in the lowest analyte or organism test cost per test, with delivering high performance results that match to the federal regulations of the FDA BAM, the AOAC Appendix J for microbial testing, and the USDA FSIS testing levels. So, in a nutshell, this new technology is effectively called a microarray. That glass slide is a DNA-based breakthrough for microbial testing that can allow labs to test a minimum of 96 samples per day, for over 50 pathogens per sample, all at the same time. So what's behind this technology? Well, as you can see on this slide, there are these wells or these little circles. And within the wells, what we have done is we printed the genetic sequences of up to 40 to 50 pathogens in these wells. And you can see these red and green dots. In this particular instance, the red dots mean normalized pathogen probes, and you can see certain green dots, and green dots represent the detection of a specific pathogen, which gives an intensity level of the amount of DNA contamination within that particular sample. And then you can see that also there's a repeatability of these dots in 3 times within the well. So what we have delivered is a ability to test a sample for whether it's E. coli or Salmonella or Aspergillus in triplicate. So what that means to the lab technician is that every sample will have 3 data points per E. coli to make sure that you, with almost near 100% confidence, that you have certainty in your result and that there's no ambiguity between a pass and a fail. And we've repeated that particular pattern across all of these wells on this glass slide so that you can test multiple samples all at the same time while testing for multiple organisms in triplicate at the same time. So at the end of the day, Pathogen DX's patented technology delivers almost unmatched performance and cost savings relative to the conventional methods that have been used for almost a century. It delivers a much faster turnaround time in the market with almost less than 6 hours of delivering results from the time you start processing the sample. In addition to that, we can test multiple pathogens at the same time down to 0.1 CFU per gram detection. We can identify the source of the contamination, as well as the economics are so advantageous, reducing the cost of testing by almost 20 to 45%. And then all of these particular organisms these tests have been built around a regulatory optimized framework to ensure that the technology is compliant to each state's microbial testing regulations. So when we take when we pause for a moment, the difference between how the world has operated in microbial testing for these organisms versus this new technology is truly the capability of multiplex testing, the ability to test for all these organisms in the same reaction at the same time, and then also being able to have cross-applicability across multiple markets, whether it's the food safety testing market or the agricultural testing market, as well as the water market, water testing market, and other markets that all require some level of microbial testing. All these organisms are pretty much similar in terms of being needed to be tested across these markets, and that's why you have the capability to apply such a technology for the benefit of simplifying microbial testing, not just in cannabis, but these other markets. The other aspect of our technology allows for the ability to extend the testing across the full supply chain. And what this means is with the technology, you can address the source of contamination where it's starting. For example, if it's in agriculture, it'll be at the farm. If it's in cannabis, it would be at the grower and cultivator level. And so what we have right now is 3 key products that are available in the market. The DetectX that allows for simply whether an organism such as E. coli is present or absent in a sample. We also are about to introduce what is called the QuantX array, which is the ability for a lab or a grower to know how much contamination is in that sample. Is that contamination level below or above the pass-fail threshold for a product to be safe or unsafe? And lastly, we have an EnviroX product which allows for an ultra-rapid capability to detect up to 50-odd pathogens, both bacterial and fungal, in an environmental setting such as a grow facility or a food processing house. Our future product plan aims to deliver a plant pathogen array, which we expect to deliver this summer, a mycotoxin array that we expect to deliver at the end of this year or early next year, a GenderX test, as well as a StrainX test. And all of these particular tests are run off of the same equipment with the same protocol and the same software. So it greatly simplifies the complexity that a lab has to deal with multiple tests. In this particular case, the resources simply are trained on one process for 7 different tests. And that's the key driver in terms of driving both predicted efficiency as well as productivity within a lab to get the results out better, faster, and cheaper. In terms of our product, this is just a pictorial view of not just the glass slides with the microarrays, but also the kit. And in this particular case, we're showing you a detect kit that allows for the detection of pathogen, bacterial and fungal pathogens. Within the kit, we provide the sample digestion buffer and a prep buffer, primers for loci enhancement, as well as labeling primers and a BSA solution, and then buffers for hybridization, as well as the 8 slides for being able to test up to 48 bacterial and fungal organisms, within each well. This particular slide basically gives a sense of how we target these particular organisms. And as you can see, there's the DetectX chip and the QuantX chip that's shown to the right. And the specific bacterial organisms that we are interrogating as part of the DetectX and QuantX, as well as the yeast and mold organisms, and specific genes as well as indicator organisms that are required by state law. Our focus is essentially to ensure that the arrays that we are developing are automated from a controls perspective. We have PCR positive and negative controls, as well as imager controls that allow for a semi-automated capability to know that the process was handled appropriately when the test was run through a lab. So it relies less on human labor and intervention and more in terms of built-in controls to ensure consistency, robustness, and repeatability of the test. I mentioned the EnviroX kit a little bit earlier. What this particular test helps is it allows you to identify a source of contamination. And as I mentioned earlier in the beginning of this presentation, up to 30% of the cannabis industry is dealing with contamination issues. So what we realized as we were developing this technology and talking to many different growers was that the actual source of contamination was within their facility, within their cultivation environment. And we developed what is called a very simple, ultra-rapid swab and result capability, where you take a swab, you basically swab it across a rack or a vent or a hood or a working table surface. And then you can submit it to your lab, and within 24 hours, have the ability to know where at those locations that you swabbed, you identified any one of these 50 pathogens that are part of this kit. And as you can see, there are quite a few organisms on the bacterial side that are basically, notorious for creating huge contamination issues within the industry. This slide is the same slide as previously, but what it also covers are some very important fungal organisms such as powdery mildew, root rot, which is Botrytis and Fusarium, as well as biofoliar organisms that can help a grower ensure that their crop is healthy and is not going to be at risk of contamination. So one of the things we did was we made sure that when we developed this technology platform and the products off of that platform, that we had a great plug and play optionality into not just cannabis, but also hemp and food and agriculture. And as you can see on this matrix, a lot of the tests that we have developed or are developing have cross applicability into the market without the need for a lot of rework, redesign, and redevelopment and revalidation. And that was the key focus of creating a building block, easy to use plug and play option for labs that don't have to have different sets of equipment for different tests, different protocols, and having to have multiple resources within the lab. And that's the key driver behind the premise of this technology, to make it a lot more easier for the labs and deliver the result much faster, better, and cheaper for the markets that they support. Now, we all know that there are other sources of other testing options, such as I mentioned Petri dish, but there is also a DNA-based method called real-time PCR. And what the difference between the Pathogen DX technology and the qPCR or real-time PCR is that the Pathogen DX technology does not require or necessarily enrichment of the sample, and that saves literally 1 or 2 days of enrichment time that other methods such as the qPCR capability requires. And so the key takeaway here is that by the time you get the result off of the Pathogen DX array or platform, you still haven't prepped the sample when you're using qPCR, and that's the difference with such a high value crop. Time is money, and it's very critical for a customer to get the results much quicker than having to wait for it while using another method that might require enrichment. The difference here is that the premise of the platform is that we have a patented approach in how we take the sample. We do not need that enrichment or incubation time. We do not need to necessarily extract, purify, or quantify the amount of DNA before you can get a result. And so that's the key game changer of the technology, as well as in qPCR, you still may be required to do what is called single analyte testing. And that means, for example, in the case of the California regulations for cannabis microbial testing, there are 6 organisms. And so each of those organisms will have to be run separately. And if you have 96 samples coming in on day one, you would need to run 96 tests for E. coli first, then for Salmonella, then for Aspergillus flavus, Fumigatus, Niger, and Terius. So it's simply a very, single plex, meaning one at a time, Adam Smith sort of, Henry Ford assembly line model relative to the PDX technology, which does multiplex testing in parallel for all organisms at the same time, while not necessarily needing enrichment or incubation of that sample. And so that's the key differentiator between the PDX technology and what is currently available in the market. So I just mentioned the ability to obviate the need for enrichment, and what's the underpinnings of this particular advantage is basically a technology called raw sample genotyping. That effectively means that you do not need to purify the DNA, quantify it, or normalize it, and no quality assessments really needed for sample contamination. All of this is avoided by avoiding almost 20 steps and/or minimum of 1 to 2 days of time through a simple 1-hour sample prep. This means that the lab will save significant time, labor, and cost in getting to the answer sooner. The second part of the process is when you've gone through the sample prep and lysis process. What we've done is we've simplified what is called the loci enhancement and labeling PCR process. For those of us that are not scientists, the loci enhancement is simply once you have basically taken your sample and you've basically cracked open the cells and identified the pathogenic DNA, the loci enhancement PCR will effectively multiply and amplify the pathogenic DNA in that sample. And then the labeling PCR is effectively applying a fluorescent marker to that pathogenic DNA so that you can effectively identify it. Essentially, this is done simplistically by having less reagents as opposed to the number of primers, the primers needed for qPCR. You only need one bacterial primer and one fungal primer for all these organisms, and that makes it a lot easier in terms of complexity management for a lab. After putting a fluorescent marker on the sample, what generally is required is that you would then take the sample and then you would basically drop it onto one of those wells, the microarrays on that glass slide. It would essentially hybridize at ambient temperature. So there's no cooling or heating requirement. It just sits there for 30 minutes. You effectively wash it, you dry it, and then you image it. And as you can see on this particular slide, you see a triplicate pattern of the dots in the center image to the right, where you see red dots as well as green dots. And as I mentioned, these particular dots represent the actual bacterial fungal organisms in terms of probes for detecting, quantifying the organism. And again, it should be mentioned that we've made the array where it's triplicate data points per organism to get a definitive pass or fail related to that sample. Once you image the data, image that well, it creates a TIFF image, and that TIFF image is effectively our software in the cloud processes that TIFF image through a dot scoring software program that then identifies which organisms were basically identified in that sample, and then delivers a result. In the case of quantification, it'll deliver a CFU equivalent data set to identify how much contamination there is related to equivalent CFUs within that sample. So when we take a step back, I've sort of described the overarching process. What we've done is when we look at the process itself in terms of equipment throughput and scalability, where we focused in terms of making sure there's no bottleneck is the imager can image up to 48 samples, in 8 minutes after scanning with real-time data reporting. So if a lab has 96 samples and in a month goes from 96 samples to 198 samples, 192 samples, all it will take is another $3,000 or $4,000 thermal cycler to process it, and not a $30,000 to $40,000 piece of equipment. So we've made it more cost affordable and capital efficient for labs to process, more samples, where the focus would be not having to spend 10 times more money for the next 100 samples. It's only incremental capital investment of another $3,000 to $4,000. As far as the equipment is concerned, there's really only 3 unique pieces of equipment. You see a microarray imager on the left. That is by far the largest piece of equipment, which is the most unique part of the process for the PDX technology. There's a $3,000 to $4,000 thermal cycler. You can see that in the middle, and an air aspiration system on the right that will draw the fluid off of the wells. Everything else is standard lab equipment that most labs have, such as multi-channel pipetters, hand vortexes, centrifuges. And so we've ensured that there's very few or very little unique pieces of equipment needed as part of this process. In terms of a layout, you do not need a clean room. You don't need a separate PCR room. All you need is 3 tables that are set up in a unidirectional flow. And so effectively it can be in a horseshoe pattern, but with a certain level of distance away from it. Not a lot of table space required. As you can see, there will only be 30 square feet of real table square footage. When you look at it as, a 3 by 6 is roughly 18 square foot. So you're really looking at three 6-foot tables set up a certain way, and really nothing beyond that. And there's not a necessary need for a hood. You can certainly use a hood if you would like, but it's not necessary as part of the equipment set. So some of you may be wondering, well, how does the Pathogen DX system compare to other technologies? And what we've shown here is basically a plate count or plate culture 3M Petrifilm technology, a medicinal genomics qPCR technology, and a bioMérieux ELISA and liquid culture technology. And at the end of the day, for labs, total test cost, which means materials, labor, overhead, and indirect, all of the indirect expenses associated with it, such as the wait time, we have provided that with respect to a sample. And as you can see, the Pathogen DX is almost 25 to 40% or more cheaper or lower cost than these other technologies. We've been able to get the level of the sensitivity and specificity of the technology much better than any of the other technologies, down to 0.1 CFU. The turnaround time is less than 6 hours versus days for the other ones, primarily because the other methods require enrichment. And then we can test for multiplex organisms, all of them at the same time in the same reaction. And then we also have inbuilt internal triplicate controls versus these other methods that have external, basically organism controls, meaning primer controls for the organisms you're testing at the end of the day. So that makes it a lot more efficient in terms of setup. For the better part of 2 to 3 decades, there's always been multiple different camps of thought as to when you bring a sample in, should you enrich or not. And effectively, for the Pathogen DX technology, enrichment is not necessary. We have conclusive validation data on cannabis matrices to show that the PDX technology has equal or better sensitivity and specificity without the need for enrichment versus other methods. And so there's always the proverbial question that a lab director may ask is to enrich or not to enrich. That is the question. Either way, the PDX technology covers that. You don't need to enrich, but you can certainly want to if you need to. But what the key takeaway here is that there are enough scientific peer-reviewed journal articles across multiple industries, whether it's the pharmaceutical industry or the food safety industry or other industries, clinical chemistry articles that have shown that enrichment biases and skews the result at the end of the day. And that's the key takeaway, is that there's enough evidence out there to say that one should not necessarily need to enrich when you're doing microbial testing, for a specific matrix. The other aspect of every lab is obviously labor. In the case of a cannabis lab, they have to run at least half a dozen tests. They've got limited resources. Labor time is scarce, and so with the PDX technology, you can run up to 96 samples with only 2 hours of real hands-on labor needed at the end of the day. And so what this particular table shows is the amount of labor time that the PDX technology saves relative to these other methods. And you can see that it's material because we've made it more semi/automated, and with the amount of hands-on time needed to run a test. So I've covered really the merits and the benefits and the process of the PDX technology. The future is now, a complete change from really how we tested compared to the past, and where PDX is now the new standard in DNA testing. So I'd like to thank you for the time and listening to me. I hope you enjoyed this conference, this presentation regarding the new DNA-based testing by PathogenDx. Thank you.
Thank you, Milan, for that informative presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that this webcast will be available for on-demand viewing through June 28, 2019. And as a final reminder, our speaker will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we'll see you next time. Goodbye.